Let me just ask you a question, Patrick. Do you know what the gram stain is used for?
I do know what the gram stain is used for.
What's the gram stain used for then, Patrick?
The gram stain is the most useful differential stain used in microbiology.
It is?
It is.
This is interesting, Patrick.
My name is Russell, and this is Joe. Joe and I are two members of our group in microbiology, and today we are going to demonstrate the proper way of gram staining.
To properly gram stain, you must first identify your slide. As you can see, we have identified our slide as group number 1. Identify the date of staining, the gram stain technique, and the the specimen species of microbes that are going to be used. Now, the first step to do is to take your slide and apply 1 drop of sterilized H2O to the slide, such as this. Next, we are going to take our inoculation loop and we are going to heat sterilize the inoculation loop with a Bunsen burner. As my lab technique Joe lights the burner, I am going to place the inoculation loop in. Allow the loop to reach a nice cherry red color. I will then pull the loop out. At this point, I'm going to let it cool for a few seconds. Coming to the Petri plate, I'm going to, as you can tell, the 2 microbes are split into A and B. I'm going to inoculate from first group A. By turning the plate over, I am going to remove some of the species, and I'm going to apply them directly to the plate. After applying them, I'm then going to heat sterilize the inoculation loop again. Allow the loop to cool. And then draw the next sample from the other side, the B slide. Once placing the samples on the slide, I'm going to do a twirling, mixing, crisscross rotation, mixing up the different gram-positive and gram-negative microbes. Finally, heat sterilizing the loop again. After adding the microbes to the plate, we now are going to dry the plate using a slide warmer, or we can allow if we wish, we can let the glass slide sit out and dry. Transferring the plate over to our slide warmer, we will wait the designated time until the slide is fully dry. After the slide has dried, we're now going to remove the slide from the slide warmer, transfer it using our clothespin. We're going to place it over the Bunsen burner in an attempt of heat fixing the microbial slides to the slide. You're going to do this by gently and slowly passing the slide over the open flame. Now, caution is the slide has now been heat fixed, and you must be cautious because the slide can be brittle and be hot. My assistant Joe will now show you guys how to properly go through and stain the slide.
The first step in the gram stain procedure involves the basic dye crystal violet, and it is the primary stain. The crystal violet stains all cells purple, and it highlights gram-positive cells. And then we can go over here. Crystal violet is applied to the heat-fixed slide for 30 seconds and then rinses with water. Here is the crystal violet that we're using. And now we will rinse the stain with water. And then we will go off to Stephanie, who will use the mordant.
Next, flood the slide with grams iodine mordant and let stand for 1 minute. The mordant increases the interaction between the bacterial cells and the primary dye so that crystal violet is more tightly bound to the gram-positive cells. After 1 minute, rinse with water for 5 seconds. Here comes my friend Patrick to tell you about the decolorizer.
Hello, I'm Patrick, and I want to tell you about the decolorizer. So, basically, we have this slide, which has already had the mordant added to it, and we're going to decolorize it with 95% isopropanol. So what we're going to do is run this over very quickly, like literally just enough to see the color run off and run clear. If you do it too much, all your cells are going to go clear, and that is a bad thing. So we're just going to do this really quickly. Okay. So now what we should be left with is the gram-positive cells being purple and the gram-negative cells being clear. And that's really important because when in the later stain, it's going to change a different color. So, but now we have to add the water to get all the stain off too, all the decolorizer off. So now we're going to wait on Sarah, and Sarah's going to come tell you about the safranin. So.
Finally, the slide is going to be stained with a counterstain. The counterstain used is safranin, which is red in color. You're going to flood the slide with safranin, and you're going to let it sit for 1 minute. The safranin will stain the colorless gram-negative bacteria pink, but does not alter the dark purple color of the gram-positive bacteria. The end result should be that the gram-positive bacteria are deep purple in color and the gram-negative bacteria are pinkish to red in color. Okay, after a minute, you're going to rinse the slide for 5 seconds. After completion of the water rinse, Sam's going to show you what you're going to do next.
Next, you're going to blot it dry on bibulous paper, but be gentle because you might crack the slide. It has been done before. It may take a couple of blots before you get it dry enough. And next, we'll take it to the microscope.
So notice she starts at 4x and she's going to work her way up using the pair focus properties, which means that she turns that, it's going to keep it in focus. So eventually she'll get up to the 100x, and that's the picture we really want to see. And she does this with the coarse focus at 4x and the fine focus at anything more than that. It really is a biology 171 concept.
Next, to see it at 100x objective, we have to do oil immersion. So we turn the objectives halfway between 40x and 100x. Then you apply the oil to the slide. There's not much left. This is not really. And then continue to slide the 100x objective onto the slide. And from here you just find focus. Okay, you can see our positive purple cells and our negative red cells.